like how Monera, what is the habitat of Monera, what is the I mean to say where they live, where they sleep, what they eat, and how they do. Okay, we all talk about it. And for today's class, we will be talking about the classification of kingdom Monera. Okay, so the Monera kingdom is totally classified into two divisions. That is, one is the Archaebacteria, and one is the Eubacteria. Okay, the kingdom Monera is divided into two divisions. That is Archaebacteria and Eubacteria. So what are these Archaea and Eubacteria? Let's take it different step for it. Okay. So the Archaebacteria are the most primitive form. Most primitive form of bacteria, and Eubacteria are the advanced bacteria. That means to say the term Eubacteria itself means true bacteria. In the last class, what we studied about the kingdom Monera actually belongs to this category. These are the true bacteria. They are the advanced bacteria, you can see. Okay. So let's talk about the cell wall composition. The cell wall of Archaebacteria is not made up of peptidoglycan. Just the Eubacteria was the cell wall of Eubacteria was made up of peptidoglycan, right? So this peptidoglycan is seen here, but in case of the Archaebacteria, okay. Also, the lipid is monolayer. We know about the bilayer structure in the plasma membrane, but it is monolayer of lipid in the plasma membrane. The plasma membrane is bilayer. Okay. Also, the lipid in the plasma membrane in this Archaebacteria, it is branched. And in case of the Eubacteria, it is unbranched. Okay. So this branch structure of the lipid that is present in the plasma membrane gives it the stability to withstand high temperature and high pH. Okay. So as we all know that Archaebacteria are they can live in the very, I mean to say, broad range of temperature tolerance and broad range of pH tolerance, that they can live in the most extreme like colder region, like from 0 degree to minus 196 degree also, and to here up to 80 to 100 degree centigrade. So they the stability in their cell wall is due to the branching of the lipid. Okay, since Eubacteria are not that much branched, the lipid is not branched, that's why they are not thermostable or pH stable. Okay. Also, in case of Archaebacteria, you will be so as soon as here that they have this 4 bacteria. Let's talk about the detailed view. Okay, let's talk about the detailed view of archaebacteria. Okay, so again, the archaebacteria is divided into three parts. One is the mechanozyte, another is the halophiles, and the third one is the hormone acidophiles. Okay, here you can see we have mechanozytes. Mechanozytes are that bacteria. They are mostly found in the marshy habitat, like the swampy habitat, all that habitat. You will find the mechanozytes. Where you will find it in the marshy, swampy area. Also, you will find mechanozytes in the ruminescent of cattle. Okay, so that is therefore in the ruminescent of cattle. You will find them. Okay, just one is also we can find them in the biogas plants. In the biogas plant, this mechanozytes are used to convert the organic material into methane. Methane gas is produced by this mechanozyte bacteria to reduce methane. Okay. Then after that they are also found in the paddy fields. Okay. As are also these are all about the mechanozytes. Let me give you some example of the mechanozytes that are mechanococcus. Mechanococcus is an example of mechanozytes. Okay. Then let's talk about the halophiles then. So what are these they love very high pH and very high temperature also. Very high pH in the sense very low pH. Okay, in pH range of 2 and heat up to 80 degrees centigrade to 100 degrees. Okay. Okay. So I have one example for you for the thermoacidophile that you will be studying in the biotechnology chapter. The example is thermos aquaticus. Okay, thermos aquaticus. This bacteria, it has an enzyme that is that polymerase that helps in the PCR chain reaction. PCR chain reaction. Why this bacteria was preferred? Because it can withstand a very high temperature. And in the PCR method also, during the time of denaturation, we will use this enzyme, this PET polymerase. It's used for the primary activity. Okay? Okay. So this is all about the archaebacteria. We talked about the mechanisms, the halophiles, and the thermoacidophiles. You have to remember this example as well. This is very important part. Okay. So now let's talk about our next part. When we talk about the archaebacteria, now let's talk about the eubacteria. Okay. The eubacteria. As the name suggests, these are the two bacteria, right? As I told you also. So, eubacteria is divided into negative two. That is the U green algae and PPLO. Okay? The eubacteria are again divided into two green algae. They are also called as the cyanobacteria and PPLO are also called as the mycoplasma. Okay? So let's talk about the cyanobacteria in detail, right? The cyanobacteria are very important according to your exam point of view. At least you can expect one question from this topic also. Okay, let's talk about cyanobacteria. This cyanobacteria is also called as blue green algae. Okay, you will ask me that we have studied about this in the algae chapter. Why are we studying this in this chapter in the Morera? So this algae actually is a prokaryote and it belongs to, it has a group under the eubacteria. So we will be studying cyanobacteria here. Okay. Okay. So the blue green algae, it has a habitat that can be fresh water or it can be marine water. It can even be fresh water as well as marine water. The cell structure, they can be unicellular structure of heterosis, it fixes atmospheric nitrogen. There is a special film that fixes the atmospheric nitrogen. The same diagram is in your NCERT book also, and this diagram is very important also. Okay, around the structure there is a mucilaginous coating. Okay, there is mucilaginous coating just outside the cell wall. Okay, this is the structure of Moster. Okay, so cell wall has mucilaginous coating. Right? The cell wall has a mucilaginous coating. Okay, one very important thing I will tell you about the cyanobacteria is it uses water for photosynthesis. You know, it uses water for photosynthesis. Rest all the bacteria we studied in the class Monera, no one was using water, and they are all aerobics. This is the first time an organism used the water for the photosynthesis process. By using the water for photosynthesis, there will be oxygen liberation. Oxygen liberation. So, it can be believed that the today's plant that are releasing oxygen mycoplasma are very interesting beings. They are also called as the joker of the plant kingdom. They can also be called as the joker of the microorganism. Joker means the microorganism. Okay. Let's talk about mycoplasma. Okay. They are called as joker of plant kingdom. Okay, they are the smallest known microorganisms. They are the smallest known microorganisms. Their size can be 0.1 to 0.3 micron. Okay. Also, they are also called as allo, that is pneumonia like organism. Why they are called as pneumonia like organism? Because this mycoplasma, they were discovered by Pasteur, and he found it the first time seen it in the bovine pneumonia, pleuropneumonia disease in the cattle. So he first time isolated in the pleuropneumonia disease in cattle. So that's why he named it as the pleuropneumonia like organisms. This mycoplasma, they do not have cell wall and lysosome. Cell wall, lysosome, flagella is also absent. Okay, they do not have cell wall, lysosome, cell wall is also absent. They can cause injury to plants and animals. Plants and animals. Okay, so mycoplasma are the very smallest organism known up to date. So if